Hello and welcome to another episode of Momento Malum today. The Adzuki bean is a cultivated species in the Vigna genus. It is consumed in large quantities in East, South, and Southeast Asian countries. China is one of the regions where the greatest level of genetic diversity. In China, over 5,000 land races and modern cultivars have been collected and conserved in the National Gene Bank of China, both for evaluation purposes, specifically for agronomic traits such as disease resistance, pest resistance, and abiotic resistance, but also for an evaluation of how much genetic variation exists within the Adzuki bean gene pool, specifically in China. Several researchers in 2 scientific institutes within China have genetically analyzed the material collected to see how much genetic diversity exists within the Chinese Adzuki bean gene pool. To do so, they analyzed a sample of the over 5,000 selections, choosing instead to evaluate 158 cultivated variations and 18 wild specimens, and analyzing them using genetic markers. A similar study has been done using another collection of Chinese Adzuki bean cultivars consisting of 435 specimens, which were analyzed based on agronomic traits rather than genetic diversity. Another study that happened the current experiment consists of a genetic analysis using SRR genetic markers, studying 176 genetic lines of Adzuki bean, which includes 18 wild relatives, 80 land races, and 78 common cultivars, commonly used as parents in the generation of new cultivars or new elite breeding line. The long list of cultivars and land races and such will be shown on the last two slides of this presentation, and the paper we're discussing will be in the description box below. Now on to the results of this paper. Through the use of genetic markers, the genetic similarity coefficient was analyzed. On the lowest end, there are strains that are 12.5% similar to each other. On the highest end, there are strains that are 93.9% genetically similar, and on average, the genetic similarity between different strains is around 54.2%. The specimens with the lowest level of genetic similarity was observed in the wild specimens, which is to be expected, and those of the highest level of genetic similarity was found in the varieties used for breeding purposes or commercial cultivars, and the land races ranked somewhere in between. Based on the genetic analysis, the sum total of all the populations analyzed could be divided up into 6 populations. The 18 wild specimens could be characterized as one population. Population 2 is found across a large and wide geography, and is likely a mixture of different populations. Population 3 seems to be derived wholly from specimens found in the Henan and Shaanxi provinces. The Adzuki beans of northern China are genetically distinct enough to be their own population. Population 5 is derived from cultivars that exist in Tianjin City and surrounding regions, and most of population 6 are derived from Hebei province within China. This indicates several breeding efforts that occurred within China, resulting in distinct genetic groups, with one population, specifically population 2, being derived from the mixture of multiple distinct genetic populations until they became their own thing. Hello, I just want to let you know, if you want to fund this channel, you can do so on the platforms seen in the description below. When comparing the genetic diversity within the wild specimens versus the cultivated specimens using the genetic markers, it was initially found that the domesticated populations had higher levels of genetic diversity in the domesticated populations versus the wild population. A result coming from the fact that it only derived from one population in the case of the wild specimens, whilst the cultivated specimens stem from multiple populations. When that was factored in, however, it was found that indeed there's higher levels of genetic diversity within the wild populations versus the domesticated populations, which is to be expected because the average Adzuki bean is found natively within China alongside other Asian countries. From a specific genetic allele perspective, there are a fair number of alleles reserved only for land races, but there's a fair number of alleles only found in the wild specimens. The researchers suggest that these may be the result of different evolutionary paths that happen through the selection process within cultivation versus what happens in the wild, and speculate that some of these traits may be useful for further breeding practices for crossbreeding the wild Adzuki beans with domesticated strains. The alleles found within domesticated Adzuki beans seem to be physically larger, carrying more DNA than their wild counterparts. This could indicate that the process of domestication led to an increase in the amount of DNA within the cultivated variation Adzuki bean. From a genome perspective, the land races, while not as genetically varied as the wild species, carry a lot more genetic variation than the modern cultivars, and can be used for further improvement of the commercial cultivars, as well as broaden the gene pool of commercial cultivars overall. Nonetheless, although the researchers were able to determine the genetic variation between populations, it was only through very fine genetic marker technology that can find smaller and smaller variations than most other technology. On a practical standpoint, there is very low variation between populations, as gene pool exchange between populations seems to happen quite a bit, and occasional natural hybridization events between both wild and domesticated Adzuki beans happens on a low level. The Beijing cultivars were found, however, to be more genetically wide, which corresponds to the phenotypic data. And that about covers everything. Now on to the cultivar list.